2 more nematodes to go. And we'll start here with a 45-year-old woman, originally from Africa, presented to her primary care provider with diarrhea, nausea, vomiting, weight loss, and gastritis. Symptoms occurred about 3 months after a kidney transplant. Stool was collected in formalin and PVA for routine O&P exam. They did a wet mount, and what you can see down here is what was found in the wet mount. So this looks really similar to other organisms that we've seen. However, I'll give you some tips to know which one it is. So take a look, pause, see what we have left, which one makes sense. And I'm hoping that you will say that this is Strongyloides stercoralis, because it is. This is a really strange organism. It causes similar GI symptoms, what we've seen before. And it has a very strange life cycle. It looks a lot like pinworm. However, the buccal cavity is different. There's no alae, remember. And what you're going to find is that in pinworm infections, you're going to see a ton of eggs, whereas it is less likely that you will see eggs in a stool sample with someone who has strongyloides. So let's jump directly to the life cycle. We have it here, and we're going to talk about this word parthenogenesis. Maybe you've heard of it before. But if we look at the life cycle, one of the things I want you to notice is that it is here where the diagnostic stage exists. It's not necessarily the egg, okay? So it's more likely that we're going to identify the rhabdidiform larvae than it is the egg. Could an egg maybe potentially show up in the stool? Sure. But it's rhabdidiform larvae that you're going to see in the stool. Now also notice what this egg looks like. It looks very similar to the egg we saw for what? Do you remember? It is indistinguishable from the hookworm eggs, Necator and Ancylostoma. So it's very tricky to tell them apart. Thankfully, you will likely not see that. Maybe we might have some slides of it, but in an actual specimen, you're going to see this. So we're going to start down here with number 1. The rhabdidiform larvae in the intestine are excreted in the stool. Now notice how there's all sorts of different paths that this one can follow, right? So if we kind of follow, I think we're going to take the path furthest to the left, that will take us furthest to the left. What will happen is the rhabdidiform larvae will live in the environment outside of the human body, and they will develop into free-living adult worms. Those free-living adult worms will then mate, you have the male and the female, and they will produce eggs that are fertilized. And when those eggs are fertilized, they will develop and hatch rhabdidiform larvae. Remember, rhabdidiform are the feeding active stage. Those rhabdidiform larvae can basically go one of two paths. They can just become adults, mate, produce more eggs, and you can have this completely independent of the human life cycle. So that's the indirect path. Or they can become the filariform larvae that are going to be infective, right? So if they become an infective filariform larvae, it is the filariform larvae that will penetrate the skin and cause infection. So kind of similar, right? We've seen this before. They will penetrate the skin and cause infection. Now notice this arrow here, right? You could have a rhabdidiform larvae in the environment, passed in the stool, that goes directly to filariform and directly infects us. So this is the direct pathway, this is the indirect pathway, and this happens outside of the body. Both of these do, actually. This is going to also be similar in terms of lung migration. So if we take a look here, the filariform larvae, because they penetrate the skin, they're going to go into the circulatory system, they're going to get into the bloodstream, they're going to get into the lungs. And once they're in the lungs, they will damage the lung tissue to get into the alveolar spaces. They are carried to the trachea and pharynx, they are swallowed, and they reach the small intestine where they become adults. Now this is where it gets really weird. In the human, in the small intestine, we're only going to find adult female worms. And somehow they lay eggs that are fertilized, essentially. But they're not necessarily hermaphroditic. It's a very strange kind of phenomenon called parthenogenesis. So parthenogenesis is when you have one sex of an organism, but they can produce, essentially what the equivalent of, fertilized eggs are, that will result in more of that organism. Bees are another thing that can do this. If you have a fertilized egg, you get one type of bee. If you have an unfertilized egg, you get another type of bee. I think the worker bees come from unfertilized eggs, and then I don't remember the other kinds of bees, but the other kinds come from fertilized eggs. It's something like that. But I know that queen bees can be parthenogenic as well. So strange. Now, these eggs that are laid by the female worm and somewhat like fertilized are deposited in the intestinal mucosa. They hatch, they migrate to the lumen, they become those rhabdidiform larvae. This is what we pass in the stool. So remember that these eggs are really being laid way high up in the gut, and they have to mature in order to pass in the stool, usually. We did not talk about this autoinfection, but the rhabdidiform larvae could just be passed in the stool and then not passed in the stool, but they could exist in the human body and mature in that lower part of the digestive tract and basically become that infective stage and recreate this, right? So this is not a liver-lung migration. The only organisms that do liver-lung migrations are going to be the ones that have to get swallowed in order for them to be ingested, right? Here we're not having swallowing or ingestion as our means of infection. We have penetration in the skin. So thinking about how they get into the body is going to tell you if they go to the liver and then the lungs or not. The only one that does liver lungs is Ascaris lumbricoides. Okay, this one does skin, blood, lungs, gut, essentially, just like we saw with hookworms. Very similar. That autoinfection, as I said before, the rhabdidiform larvae in the large intestine, they become filariform larvae. They penetrate the mucosa. They get through or the perianal skin if they made it all the way down, out of the gut. And then they basically just do that same pathway over and over again. There's many, many, many of them, right? There's a lot of ways this thing can be infective. It's kind of strange. You would expect to see GI symptoms and cutaneous symptoms, right? As it's penetrating the skin. So that kind of itching, right? And also pulmonary symptoms. So very similar to what we've seen before. We've seen a lot of similar entryways into the human body. But that parthenogenesis thing is really weird. And I'm not doing it justice describing it. But basically it's saying you can produce more organisms without having the male there to fertilize the eggs. That's parthenogenesis. And you can read more about it if you want. I'm not going to go into a lot of detail on it. So our last worm, you do not have to know the life cycle of, but I do want to talk about it. It's very interesting. It is Dracunculus medinensis, also known as the Guinea worm, and it is big and strange. It is very rare to see the male. The female has a very blunt, round anterior end. You will actually see the adult, and you can read about how it has to be removed. So what you're looking at here, this is someone who's infected with the guinea worm, and what happens is the female gets into kind of the tissues and essentially gets its way down to the foot where it creates this boil, right? This blister. And this blister doesn't feel good. And a lot of times what you will do to kind of remediate the symptoms, because it feels kind of hot, is you'll put your foot in water, and that will actually cause the blister to rupture. And that rupturing is exactly what the parasite wants you to do, because what the worm is going to do is it's then going to stick its uterus outside of this burst ulcer at this point and spew a bunch of larvae into the water. This is contracted through drinking dirty water. And what you will find is that once that happens, you're just perpetuating the life cycle. So this worm is incredible because what it does is it basically creates a situation where you will go and find a water source to alleviate symptoms, and that is the next step of its life cycle. It is mind-blowing. Now, if you want to get this thing out, it's tricky because it can run the length of your leg, honestly. It will, if it breaks, if you want to just pull it out, if it breaks, it can actually cause some really severe allergic reactions and very toxic if you remove it and it breaks. So what you have to do is you have to take the worm and tie it around, this is a match you can see here, but a little stick. And that little stick, basically every day you just wrap a little bit more of that worm around that stick, and then you wait. And then the next day you wrap a little bit more. You can't just wrap the whole thing, because if it breaks again, there's going to be like very toxic side effects. So you wrap a little bit more, and eventually you get the whole worm out. It's very interesting. Now, as I said, this is something that is contracted through drinking water. So if we kind of look at how this goes, you have a human who drinks unfiltered water, and what you're going to get in that unfiltered water are copepods, which we've seen before, that have a larval stage of dracunculus in them. The larvae, when you eat them, are released. When the copepods die, they penetrate the stomach and the intestinal wall. They mature and they reproduce. Look at the difference in the female and the male. It is very rare that you're going to see the male. It's like the tiniest little thing, and the female is this big long worm. So what will happen is the fertilized female worm will migrate to the surface of the skin, which is essentially down here, kind of goes through the subcutaneous tissues, and these little red lines are showing it migrating from the intestinal mucosa down here, where it causes that blister. And when the blister bursts, the larvae are released, so it lays live larvae, which is also kind of unique. The female worm it takes about a year for that emergence to happen, and this is a diagnostic stage, is seeing that worm from that blister. The larvae that are laid will go into the water source. They will be eaten by a copepod. They will continue to develop into what is known as an L3 larvae. You don't have to know that, but know that there's developmental stages that happen, and then the human drinks the water. So pretty wild life cycle, and like probably the craziest one to, I don't know, we'll see. There's a question on your quiz about which one you'd want the least. This would probably be mine. Now, one of the coolest things that has happened in the last, I don't know how many years, but we have done a really, really good job of getting the level of this organism, the infection rate, way down. And I think former President Jimmy Carter had a big hand in that. Basically, they have developed straws that will, when you drink through them, it filters out the copepods and prevents you from intaking, that larval stage. Also, there's been a big push in teaching people how to just filter the water, just like filter it through a cloth. It doesn't even have to be something super fancy to help prevent this. So as you can imagine, having something like this, having that ulcer is very limiting, right? Because one, it's an open wound, and if you're someone who has to work, it's going to hurt. It's also kind of, there's a stigma around it, having these sores and these worms. And so it can be really problematic for lots of reasons besides just the fact that you have a worm sticking out of a hole in your foot, right? So getting rid of this in developing countries, typically in areas where, water sources are not as sanitized, has been really amazing and beneficial. And you can look and see the number of, the incidence of infection is so low now, just from that push to get the filtration up to par, that it's very minimal how much we see of this, which is awesome. So you can dig into that too if you feel like you want to. I don't need to talk about it anymore. So this wraps up our discussion on the nematodes. We're done pretty much with the worms. We'll see, there's kind of a wormy thing to look at later. But if you have questions, you know how to get a hold of me. Let me know.